Hello everyone, my name is Giuseppe Balsamo and today I will be talking to you about the separation method known as centrifugation. So, what is centrifugation? Centrifugation is a process by which particles are separated from their surrounding mediums through the use of centrifugal forces, or G-forces. This is usually done through the use of a centrifuge machine. Centrifugation is also a popular process used during biopharmaceutical research. So, how does it work? The process of particles separating from their surrounding mediums can happen naturally, for example, like with sand in the ocean or snow in a snow glove. However, within the biopharmaceutical field, many substances that are tested will not separate naturally, or at least not in a fast enough manner. This is when centrifugation comes into place. Centrifugation speeds up this process through the use of a spinning rotor in a centrifuge machine. The rotor creates g-forces, which accelerates the speed at which substances separate into its relative parts. This process forces separation to occur at rates thousands of times faster than would occur naturally. Following this image right here, you'll see a test tube. And this test tube will usually what you see people placing inside some common types of lab centrifuges. And I'm going to talk to you about a little bit of what the results of the centrifugation will look like. So when a substance is spun in a centrifuge machine, the denser component of the substance being tested, known as a precipitate, will move away from the axis of rotation. The less dense component, which is known as the supernatant liquid, will then move towards the axis of rotation. Again, in this diagram shown right here, the test tube was placed in a centrifuge machine with the bottom of the test tube placed further from the axis of rotation than the top. Therefore, the precipitate is at the bottom, while the supernatant liquid is at the top. The precipitate or supernatant liquid, depending on which one we're testing for, can then be extracted to be used for research or medical purposes. There are two main types of centrifuge. One is the sedimentation centrifuge and the filtration centrifuge. Each type separates a denser precipitate from a less dense supernatant liquid. However, the methods of separation and extraction differ a little bit from each of them. So, sedimentation centrifuge. In a sedimentation centrifuge, the precipitate, or the denser component of substance, sticks to the wall of that centrifuge machine, while the supernatant liquid, or less dense component, overflows and gets removed by a skimmer. The precipitate can then be extracted manually with a cutter knife or screw conveyor. Now, what about filtration centrifuge? Filtration centrifuge, in this, the machine contains an internal basket filter. In this case, the centrifuge spins and the supernatant liquid is expelled through the filter from the rotations or spinning motion, while the precipitate gets trapped within the filter. As you basically can see in this picture right here, as the rotor in the centrifuge machine is spinning, there's basically a filter there's basically a filter. The porous walls of the filter are small enough for the less dense components to pass through, but they are too small for the denser component to pass through, which causes the separation. After this process is completed, the precipitate can be extracted from inside the filter. Besides the two main categories of centrifuge that we just talked about, there also are some subcategories of centrifuge. Some of these, for example, are test tube centrifuge, ultra centrifuge, tubular bowl centrifuge, amongst many others. Different types of centrifuge cater to different needs. For example, a tubular bowl centrifuge clarify heavily contaminated liquids in an early purification phases. Decanter centrifuge manage large volumes of suspended solids, and ultra centrifuge isolates biomolecules and nanoparticles with precision. Again, just depending on what you're expecting to extract from a substance, you might have to use a specific type of centrifuge to achieve your desired result. Now, in continuation, we'll talk about some examples in regards to centrifugation. So my first example has to do with one of the most common types of applications on centrifuge within the biopharmaceutical field. This is the removal of plasma from the red blood cells in the blood. So to usually facilitate the separation of plasma from red blood cells, blood is placed in a capillary tube, which is a very thin test tube, and then sealed. These test tubes are placed in a test tube centrifuge and are spun for usually between 4 to 5 minutes until the plasma splits from the red blood cells. As the diagram shows, the red blood cells sinks to the bottom while the plasma rises to the top. In this case, the red blood cells will be referred as a precipitate since they are the denser component, while the plasma will be referred to as a supernatant liquid as they're the less dense component. The resultant plasma can then be used for medical purposes or measured to see if a patient blood plasma levels are low. Another example, another example of like how centrifuges are used is for the development of vaccines. For instances, when spun at high speeds, a continuous flow ultracentrifuge machine can be used to separate or purify a virus or antigen. This purified virus or antigen can then be used to develop a vaccine to fight that type of infection disease. As you can see in the image right here, we have an example of a continuous flow centrifuge in which basically a density gradient is formed inside the rotor. This causes the target material to separate out in the density gradient band from which it can then be removed. Lastly, my last example has to do how centrifuges can be used to isolate substances, such as protein from cells for extraction. In this instance, a sample of cells is placed in a density gradient ultracentrifuge and spun until the various components of different densities are separated out. Further centrifugation cycles can then be used to eliminate undesirable cellular impurities or to obtain certain cell organelles, such as proteins. This process is usually quite useful for research as proteins are an important biomarker for diagnosing diseases. So what are some pros and cons in regards to centrifugation? In regards to the pros, as I mentioned before, one of the biggest advantages of centrifugation is the ability to speed up these separation processes. As I mentioned, these machines allow the separation of substances to occur much faster than what happens naturally to gravity. Note that centrifuge machines commonly operate at speeds between 500 to 20,000 rotations per minute, or RPMs. This is incredibly helpful in lab settings where tests need to be run quickly and efficiently, often many at a time. Centrifuge machines are also incredibly versatile and can be modified to complete specific and unique lab functions. These include everything from separating or concentrating substances to filtering or rinsing substances. Again, this is as we talk about how they're within the two main types of centrifuge machines, there are their different subtypes. These machines are also relatively safe and easy to use. Usually, once left to run, a centrifuge machine can be left alone until samples are ready for collection. Centrifuge machines also contain a lot of safety features to prevent them from injuring the user or from leaking. This is important as dangerous and biohazard substances, such as blood, are usually spun in a centrifuge machine, which can be dangerous if spilled. Now, in regards to the cons of centrifuge machines, one of the main issues with centrifuge machines can be price. Usually within lab settings, lab centrifuge can easily cost between 2,000 and 7,000. All this can be depend on the model, make, and brand of such. Of course, like depending on like what they're needed for and what necessities are, and for the lab or outside of lab, centrifuges can run exponentially much better than that. While some highly funded labs or hospitals can afford this, others cannot. Centrifuge machines are also generate a lot of spinning, which can be distracting for those nearby. This can also make it difficult to focus. Again, this type of noise, depending on how you have it, like where you have it placed, could cause like hazardous situations. Finally, while it's relatively safe for some, well, they're relatively safe to use and to be left alone spinning, centrifuge machines can produce a lot of heat. This can be very dangerous if proper steps and safety equipment are not used when handling a centrifuge machine. Now, overall, centrifuge machines are very popular and a very successful method of separation used within the biopharmaceutical field. Centrifuge machines are customizable to fit a variety of functions and make a slow process of separation occur very quickly. They help with research, such as vaccine development, and with medical testing, like blood plasma level tests. And while there are some cons to the machines, such as price, noise, heat, the pros usually heavily outweigh this with their speed, safety, and ease of use. Finally, here are my sources used for this presentation. Thank you so much.